Topic DNA, The Code of Life Lesson 2. Aims of lesson. At the end of this lesson you should be able to know the following. Structure and role of DNA. Replication of DNA. DNA profiling. Introduction. Revise the location and history of the discovery of DNA. Revise nucleic acids that were covered in grade 10. Concepts and skills. Structure of DNA. DNA is a polymer and consists of two strands that are twisted to form a double helix. The monomers of DNA are known as nucleotides. Each nucleotide consists of a sugar molecule, deoxyribose, a phosphate group and a nitrogenous base. Deoxyribose combines with the phosphate group. One nitrogenous base combines with deoxyribose. The sides of the DNA ladder consist of alternating deoxyribose molecules and phosphate groups. There are four types of nitrogenous bases in DNA, i.e. adenine, A, guanine, G, cytosine, C, and thymine, T. Adenine always combines to thymine. Guanine always combines to cytosine. There are equal numbers of adenine and thymine bases as well as equal numbers of guanine and cytosine bases in a DNA molecule. A equals T and G equals C. Nitrogenous bases are joined by weak hydrogen bonds which are easily broken by enzyme action. Know the meaning of instructional verbs in test and examination questions, e.g. Instructional verb. Meaning. Describe. State in sentences the main points of a process. Explain. Give your answer in a cause-effect or statement and reason sequence. Compare. Give similarities and differences between concepts. Question 1. 1. The diagram below represents a portion of a DNA molecule. June 2019 P2. 1.1 Identify parts B, C, and D. 1.2 Name. A. Monomer A. B. One organelle in a cell where DNA is located. One DNA strand is the complement of the other strand. Functions of DNA. Sections of DNA forming genes carry hereditary information. DNA contains coded information for protein synthesis. DNA replication. DNA replication is the process during which a DNA molecule makes an exact copy, replica, of itself. DNA replication takes place during interphase of the cell cycle. 1. The DNA double helix unwinds. 2. Weak hydrogen bonds between nitrogenous bases break and the two DNA strands unzip. 3. Both DNA strands serves as templates. 4. Free nucleotides in the nucleoplasm are used to build a complementary DNA strand onto each of the original DNA strands, A to T and G to C. 5. This results in two identical DNA molecules. Each molecule consists of one original and one new strand. Significance of DNA replication. Doubles the genetic material so that each cell receives the same amount of DNA during cell division. Results in the formation of identical daughter cells during mitosis. DNA profiling. Every person except identical twins has his own unique DNA profile. It can be described as an arrangement of black bars representing DNA fragments of the person. DNA profiling can be used for the following. As proof of paternity. To trace missing persons. To identify genetic disorders. To establish family relations. To match tissues for organ transplants. To identify dead persons/animals. To identify crime suspects in forensic investigations. Common errors made by learners in examinations. Unable to answer and interpret questions based on the structure of DNA, DNA replication and DNA profiling. Unable to make appropriate drawings and label diagrams. Activities/assessment. Complete the questions in this lesson. Consolidation. Work through the questions on the structure of DNA, DNA replication and DNA profiling in past examination papers. Reflect on your learning and understanding of this topic and assess your progress. Values. By studying this section, you will develop an ability to critically evaluate and debate scientific issues, concepts, and processes. Thanks for watching. Please do not forget to like, share, and most important, subscribe to the channel.